So our lecture today will be about cortical spreading depression. Now, what is cortical spreading depression? Really, we hear a lot about it when it comes to the pathophysiology of migraine, but we just accept it and move on. But let's talk about it.
More than a headache, it's a neurological storm. Phases that shift and change form. Aura the pain, it's a complex disease. Migraine is a journey, not just a squeeze.
Cortical spreading depression is a phenomenon in migraine that is usually associated with the aura phase of migraine. When it comes to aura, we have our typical aura, and we have our non-typical aura symptoms. Now, what's typical and what's non-typical? Well, let's follow along with unraveling migraine. So our typical aura symptoms are visual, sensory, speech, and or language. Those are our three typical aura symptoms. And they generally move in this order as well. Not always, but usually. Then we have our non-typical aura symptoms. This is motor. We see this in hemiplegic migraine. All our non-typical aura symptoms have their own diagnosis. It's not just simply migraine. Brainstem, we see this in migraine with brainstem aura, and retinal, and we see this in retinal migraine. Not only does cortical spreading depression result in aura, or is the mechanism behind aura symptoms, but it also results in pain involved in migraine. And we can see this, again, in unraveling migraine. Under migraine pathophysiology, we see trigeminal vascular system activation. Cortical spreading depression activates trigeminal nerve afferents innervating meningeal blood vessels. And according to new research, it also leads to changes in CSF composition. Now, these changes include elevated proteins or elevation in certain proteins. And one of the most common ones we know that is involved with migraine pain is CGRP. So cortical spreading depression actually causes changes in CSF composition, increasing proteins like CGRP that then find its way into the trigeminal ganglion, thereby causing the pain. Now, to understand cortical spreading depression, let's break it down word by word. So let's start with cortical. Let's just get this erased here. So what does cortical mean? Cortical means the cerebral cortex, the outermost layer of the brain. Now, if this were a 3D image, it would be the entire outermost layer of the brain. But if we look at it as more of a sagittal cut, we see that it's this outermost layer right here. Gray matter. Now, this is important for a few reasons, and one of the reasons is location. So cortical spreading depression usually, but not always, starts at the occipital lobe. Okay, so let's actually label this end of the brain as the posterior end, and this as the anterior. Let's just do A for anterior. Cortical spreading depression usually starts at the posterior end of the brain, where the visual cortex is. It's always important to ask ourselves why. We know that migraine aura almost always starts with a visual aura, and it may also be the only aura present, and it's also the most common type of aura in migraine with aura. But why does this occur? Why can't it start with a language aura like we talked about? Albeit sometimes it does, but not usually. So why the occipital lobe? Well, it's because the occipital lobe has the highest neuronal density in the cortex. It makes it more susceptible to cortical spreading depression. Higher neuronal density means more tightly packed neurons, which is the perfect environment for the spread of electrical and chemical changes that are characteristic of cortical spreading depression. Because cortical spreading depression is a self-propagating wave of neuronal and glial depolarization that spreads across the cerebral cortex at a rate of 2 to 6 or 2 to 5, depending on which source you read, millimeters per minute. So it's a wave of neuronal and glial depolarization. I understand you don't know what depolarization is yet, but we will get to it. You will understand by the end of this lecture. So, like we talked about, this wave generally starts from the posterior end of the brain and moves anteriorly, from the posterior to the anterior. Again, occipital lobe has the highest neuronal density, and we will get to this, but cortical spreading depression can also be started due to the increase of neuronal activity. Another reason why it makes sense why higher neuronal density will result in cortical spreading depression, and therefore a visual aura is most likely the first symptom of aura and possibly the only symptom. Now, as it progresses from posterior to anterior with migraine with aura, we see different symptoms occurring. So, for example, again, in the occipital lobe, let's just label this occipital, we see changes in visual processing. Why? Because the occipital lobe is in charge of visual processing. So when we see migraine with aura, we see visual aura. Now, as it moves posterior to anterior, let's actually draw the central sulcus here so we don't get confused. There you go. And then we have our parietal lobe. So this would be our parietal lobe. And when it goes posterior to anterior, what was the next aura that we usually see? We see a sensory aura. So this would be things like paresthesia. Again, we'll get to this in more detail a little bit later. So parietal lobe, and the parietal lobe is in charge of sensory processing. So this will be sensory aura. Now, as it continues to progress, we have the temporal lobe here. And we have the frontal as well. Now, the frontal lobe is in charge of motor control. However, there's also a location in the frontal lobe that's in charge of language. So as it progresses from posterior to anterior, there are locations in the frontal lobe and in the temporal lobe that are in charge of language. So temporal, language aura, and frontal also is the language aura. But also, with hemiplegic migraine, we have a non-typical aura. So when it goes to the frontal lobe and the primary motor cortex, we have motor aura. Okay, this is non-typical. The rest are typical aura symptoms. So visual aura, we start with visual aura, then sensory, usually, and then language third. So again, as CSD, cortical spreading depression, progresses from posterior to anterior, there are an array of symptoms that can occur. Now, not only is the location, the cortical part of cortical spreading depression, not only is that important, but also the structure of the cortex is important too. What's in the cortex? The cortex is gray matter. Okay, so let's just put structure. So structure, the cortex is gray matter. That means there are neurons, well, specific parts of a neuron, and glial cells in this gray matter, and both of which are affected by cortical spreading depression. So glial cells, particularly astrocytes, play important roles in maintaining ion balance and neurotransmitter levels in the extracellular space. So during cortical spreading depression, both of these cell types are impacted, neurons and glial cells, particularly the astrocytes. So that's why the cortical part of cortical spreading depression is important. All right, let's erase this so we can move on to our next topic so we can better understand cortical spreading depression. Now, to better understand cortical spreading depression, I want to compare it to a normal way our body sends electrical signals. And it does this via an action potential. Action potential. That's how a neuron is able to propagate an electrical signal. It's how I tell my hand to move right now. It's a three-order neuron system that communicates with each other in order for me to move my hand. Now, this action potential is an electrical signal. This is how a neuron is able to propagate a signal. It is with the action potential. So let's draw a graph here. This will be your membrane potential, and this will be time. Now, time here will be in milliseconds. We'll change that a bit when it comes to cortical spreading depression, and you'll know why later. And the y-axis, of course, is the membrane potential. Now, neurons have a resting membrane potential of -70. This is the resting membrane potential of -70 millivolts. And this is maintained, so this is the resting membrane potential. And this is the neuron, okay? This is maintained by the sodium-potassium ATPase pump. By doing this, it allows 2 sodium to go in, let's say this is the neuron here, for every 3 potassium that comes out. So you have more positive coming out than positive going in, and therefore you have a negative membrane potential, okay? Also, there's leak channels involved, but let's keep it simple for this lesson on cortical spreading depression. Now, the neuron isn't doing anything here, right? It's just, again, it's resting membrane potential. There's no signal. It has to reach a specific threshold. Action potentials require a certain threshold to be reached before depolarization, which is that word again that I told you we're going to get to. So it has to reach a specific threshold. And normally, that threshold is about -55 millivolts. So the neuron is stimulated. Now, the way it's stimulated is actually different between cortical spreading depression and the specialized ability of a neuron to send a signal via an action potential, but we will get to that in a bit. So the neuron is stimulated and the cell becomes more positive. Why? Because sodium is positive. So you have more sodium rushing in, the cell is becoming more positive. And once it reaches the threshold, it's an all or nothing response, okay? All or nothing. So if it doesn't reach the threshold, you have nothing. There's no small response. It goes the entire way. And so this is sodium going in. Don't think of it as decreasing, sodium rushing in, okay? And when it reaches the threshold, voltage-gated sodium channels open. Let's actually change this so it's not confusing and just let's call it influx. We have sodium influx. And usually the peak it reaches with depolarization is about +40 millivolts. +40 millivolts, okay? So this is depolarization. This is called depolarization. This is what actually moves the signal. This is how the neurons communicate. And then we have our repolarization. With our repolarization, sodium channels close, potassium channels open, so sodium channels close, and potassium channels open, causing a potassium efflux. Okay? Potassium is late to the party, it opens late, the efflux starts late, and that's why we have repolarization. However, because they're so slow, we actually have a hyperpolarization before it finally goes back to the resting membrane potential. So again, potassium flows out, cell goes back, a neuron is a cell, okay, it's a nerve cell, eventually goes back to its resting membrane potential. Again, hyperpolarization is just because it's a brief overshoot, like it's -70 here, it probably reaches -75 millivolts, because the potassium channels take a while to close. Now let's talk about this recovery. How does it recover? Well, the same way it maintains the resting membrane potential, via the sodium-potassium ATPase. Now this pump is an active pump. Active meaning it's not passive, it requires energy, it requires ATP to function. And this becomes important with cortical spreading depression. So the sodium-potassium pump restores the balance here. And that's what we need. We need normalcy again. Now let's contrast this process to what happens with cortical spreading depression. Let's draw another chart here. And this will be the membrane potential again, but so the y-axis is the same, right? It's in millivolts. However, the x-axis, instead of milliseconds, the time will be in minutes. This represents how long it takes for cortical spreading depression to move across the brain. And it also represents how long an aura lasts. So the first thing, Triggering. An action potential is triggered by a specific stimulus in a localized part of the neuron, usually the soma or the cell body. So let's say this is a neuron, and we have dendrites here, and then this is the axon, and this is the axon terminal. So an action potential responds to a stimulus in the dendrites receiving the information, and then eventually when it's done, when the action potential propagates across the axon, reaches the axon terminal, and neurotransmitters are released at the axon terminal, okay, part of the synapse. Now, when it comes to cortical spreading depression, it can be triggered by various factors. With cortical spreading depression, it could be triggered by an increase of extracellular potassium. Now, we aren't talking about serum or blood potassium. Please don't think that when we talk about ion changes here, it is referring to changes in the blood. These are only local changes that are necessary to send to propagate this electrical signal. So, high extracellular potassium or intense neuronal activity. And we talked about this already because that's why the occipital lobe with the highest neuronal density is likely the first to experience cortical spreading depression, and it may be the only aura symptom. So, heightened neuronal activity or an increase of extracellular potassium. This is very different than the highly specialized way neurons normally communicate with each other. Now, let's erase this here and talk about why. By the way, these are the triggers we know about in regards to what gets cortical spreading depression started, but there are likely many, many more. But why these various triggers? Well, the cortex, again, because we know it happens in the cortex, right? Cortex is complex, and disruptions in ion balance or excessive neuronal activity will result in creating this tipping point. So these small changes of increased neuronal activity will make the cerebral cortex reach a tipping point where normal regulatory mechanisms are overwhelmed and thus initiating cortical spreading depression. So many things, more than a highly specialized neuron, highly specialized action potential of a neuron can make it reach the threshold in order to result in cortical spreading depression. Again, this is for cortical spreading depression, okay, millivolts. Now let's talk about depolarization. Let me just fill this in here because it got erased. And for cortical spreading depression, the depolarization is massive over a large area, but it doesn't reach plus 40 like a normal neuron. What it reaches is 0 millivolts. And thankfully, because we can't have this wave of depolarization acting like a neuronal signal. So there's controlled depolarization reaching 0 millivolts, but it's prolonged. Prolonged over a wider area, but not as intense as an action potential that reaches plus 40. And again, action potential travels along a single neuron at a time, high speed, right? And that's why the x-axis is different on both of these. Time here is in milliseconds. Here is in minutes. Huge difference. So while this travels, let's say, at around 100 meters per second, depending on the type of neuron, cortical spreading depression slowly goes across the cortex from posterior to anterior at a rate of about 2 to 5 or 2 to 6, depending on the source you read, millimeters per minute. And while it does this, it affects multiple neurons and glial cells. So a much, much slower process. An action potential lasts a few milliseconds. Depolarization can last a minute or more. And this is followed, again, don't confuse depolarization with depression. The D in cortical spreading depression. Depolarization just means the cell is depolarizing, or when we talk about neuron, or the area that we're referring to in the cerebral cortex is depolarizing. So for example, let's say, like, how do we measure depolarization, for instance? So there's an electrode inside the neuron. Okay, this is not an arrow, it's an electrode. Just assume it's an electrode. And it's connected to a voltmeter. And we also have the ground here, okay? Now the voltmeter will read the voltage. So when the neuron is undergoing depolarization, it'll read plus 40. So we can actually objectively measure this. Now when we come to cortical spreading depression, Because it happens in the cortex, we're not going to stick an electrode intracellularly, but there is a strip we can use that has 3 electrodes, put it on the surface of the brain, have that connected to a voltmeter, same way, and then have another ground. So don't confuse the depolarization, which is the measurement we see here, with depression, which we didn't talk about yet. So this depolarization phase lasts about a minute or more, and then it's followed by a long depression. You see how it's followed by a long depression? Let me actually put it on the screen here, even though it's long. Just assume it goes past it. So we have depolarization here, but depression here. Now, why does it take such a long time? Well, there are massive disturbances in cellular function, and it takes time to resolve. The brain needs to recover from this massive and lengthy cortical spreading depression disturbance. So we see here, there's a quick recovery, right? Milliseconds. There's a quick recovery to resting membrane potential. But with cortical spreading depression, we have a prolonged recovery. And we see this in the symptoms of migraine. There is a prolonged recovery. But why does that occur? Well, the sodium-potassium pump is in every cell. It's also in the cerebral the neurons in the cerebral cortex, right? So the way it will get back to, the cortex will get back to its resting membrane potential is also the sodium-potassium pump, which requires energy. So it requires such a massive energy demand, even though it only depolarizes to 0 millivolts, it requires so much more of an energy demand due to the spread of cortical spreading depression, due to the massive ionic imbalances that occur in the cortex. It requires such a massive amount of energy in order to reach the resting membrane potential again. And this means that you need large amounts of ATP in order for ions to move against their concentration gradients so we can reach the resting membrane potential again. Now, this results in an increase of up to 250% increase in glucose use. So this is a massive energy demand that is required. So it kind of makes sense. This may be part of the reason we're fatigued after a migraine attack. So much energy has been used. Very different than normal neuronal firing. And here we have controlled localized changes in sodium and potassium concentrations. In cortical spreading depression, we have these dramatic shifts in ion concentrations across a large area where extracellular potassium can reach 20 times the normal limit. And also within action potential, you have this controlled release of neurotransmitters. When cortical spreading depression occurs, it's a massive widespread release of neurotransmitters like the excitatory neurotransmitter glutamate. It's massive. It's very controlled, very specific, very action specific. When I tell my hand to move forward, it's not moving everywhere. It's not spasming. There's no fasciculations. It's doing what it's supposed to do because it's very controlled. Cortical spreading depression is not controlled. It only affects neurons. Cortical spreading depression affects neurons and glial cells. So we have something that's completely essential for normal brain function and communication, and something that disrupts normal brain function. It leads to migraine aura symptoms and potentially contributing to pain. Now, this prolonged depolarization, again, is followed by depression. This is when the neurons shut down. They're not functioning normally anymore. And this also manifests as various symptoms. For instance, you can have, and we'll get to this, you can have positive symptoms, and then you'll see negative symptoms like blind spots, scotomas. And this moves as this wave progresses anteriorly. So while one area can be depolarizing, another area can be in the depressing or the depression phase. Again, depolarizing is considered active and depression is considered inactive. Now, we talked a lot about cortical, and we talked a lot about spreading, right? We know, for instance, that it spreads at a rate of 2 to 5 millimeters per minute. Again, slower than action potential. The entire duration can last up to an hour or more with cortical spreading depression. It depends on how far it spreads and how long it takes. Let's actually erase this. So we know as cortical spreading depression moves, it affects different areas of the brain. So therefore, we have different symptoms like we talked about before. Let's see if it's wet. It's still a little wet, but let's move on. So when it starts in the occipital lobe, we have visual aura. What does that entail? Well, flickering lights, also known as scintillations. We can have zigzag lines, also known as fortification spectra, or we can have blind spots, also known as a scotoma. When it passes through the parietal lobe, we have sensory symptoms. With sensory symptoms, we have paresthesias, also known as pins and needles sensation, tingling, anything, any sensory symptom. When it goes through the temporal or frontal lobes, we have language aura. And this could include trouble finding words, speech problems, dysphagia or aphasia. This is all caused by cortical spreading depression. Again, these are typical aura symptoms. We also have non-typical. So if it impacts the motor cortex, we will have hemiplegic migraine, and that is temporary weakness on one side of the body. If it happens in the brainstem regions, it may contribute to vestibular migraine, resulting in vertigo and dizziness, or even another type of non-typical aura called brainstem aura. However, we actually don't know if this is due to CSD in the brainstem. It's kind of debated right now. And when it comes to retinal migraine, another non-typical aura, spreading depression of retinal neurons may occur. This is one theory anyway. And that's similar to cortical spreading depression in migraine with aura. So this is migraine with aura, and you would have a different diagnosis if the cortical spreading depression was in the primary motor cortex, for instance, and there's weakness on one side, and then you would have hemiplegic migraine, right? So it all comes back to cortical spreading depression. So the symptoms reflect the functions of the cortical areas affected as this wave propagates, moving from posterior to anterior. Now let's talk a little bit more about depression. So after this heightened activity, this depolarization, right, we have depression. And this is where the affected areas of the brain can't function. And this can also explain the progression from positive symptoms like scintillations and fortification spectra to symptoms like blind spots that occur. Sometimes the flashing lights move laterally and the blind spot follows it. That is the depression phase. And what's interesting about the depression phase, it's likely a protective mechanism to prevent further excitotoxicity and allow for metabolic recovery, because we don't want to keep the cerebral cortex firing. Now, there are a few things that are also worth mentioning. That cortical spreading depression isn't only about just these symptoms, but there are also blood flow changes. So hyperemia, there's an increase in blood flow and then a decrease in blood flow after. So transient increase in cerebral blood flow followed by a prolonged decrease in cerebral blood flow. And the cerebral blood flow, so increase and then a decrease. And this decrease can last longer than the depression, last longer than the electrical activity suppression. Furthermore, there is an increase of reactive oxygen species, an increase of oxidative stress. So while cortical spreading depression doesn't cause permanent damage in a healthy brain, the recovery process is demanding, and it can contribute, like I said before, to the fatigue that one can experience after the headache phase or even during. Again, the brain has to restore normal ion gradients. It has to clear excess neurotransmitters. It has to repair the cellular damage that occurred. And it has to replenish the energy that was taken up from this entire process. This requires significant time and significant resources. Also, what's interesting about cortical spreading depression, there's also evidence that if it impacts the hypothalamus, for instance, it can result in yawning. Where do we see yawning? We see yawning in the prodrome phase of migraine. We see yawning in the prodrome phase. So cortical spreading depression may also result in some of our prodrome symptoms, like yawning, like food cravings, like mood changes. The hypothalamus regulates autonomic functions and neuroendocrine processes. So disruption here could explain these diverse prodrome symptoms. So cortical spreading depression may actually be happening before the aura phase, potentially. If it reaches the thalamus, it could explain the heightened photophobia, which is sensitivity to light. It could explain the phonophobia, which is the increased sensitivity to sound. If it's in the brainstem, nausea, vomiting, dizziness, disruptions of other autonomic functions, amygdala, anxiety or emotional changes. So cortical spreading depression can affect the prodrome phase, the aura phase, and the headache phase, and the postdrome phase. It's an important part of migraine pathophysiology. Migraine is a neurological disease because phenomenons like this are the underlying pathophysiology of migraine.